The overall goal of this video is to show how to fabricate and use microfluidic chips with integrated electrodes for transendothelial or transepithelial electrical resistance measurements, or TEER measurements. This is demonstrated using a blood-brain barrier in a microfluidic chip.
This method can help answer key questions in the field of organs on chips by enabling direct measurement of the barrier function of, for example, blood-brain barrier tissue using integrated electrodes.
The main advantage of our technique is that the electrodes are easily integrated into organ-on-chip systems and that the resulted TEER values can be compared among different systems.
The implications of this technology extend towards understanding blood-brain barrier function in health and disease, drug discovery, and personalized medicine. Though this method can be used to provide insight into blood-brain barrier function, it can also be used in the context of other organ-on-chip systems, such as the lung-on-chip and the gut-on-chip.
To begin this procedure, mix 27 grams of PDMS base agent and 2.7 grams of curing agent thoroughly. Then, degas the mixture in a desiccator for approximately 45 minutes to remove air bubbles. Meanwhile, prepare the mold for the liquid PDMS mixture by sticking clear tape around the mold or place the mold in a suitable wafer holder. Pour the degassed PDMS mixture onto the mold. Next, cure the PDMS mixture in an oven at 60 degrees Celsius for 4 hours and allow it to cool down afterward. In a cross-flow hood, pull the cured PDMS from the mold. Cut the PDMS replica into separate top and bottom chip parts using cutting lines in the PDMS.